welcome to 17 Minutes of Science, our show that explores the world of science and how it affects both the starting academic and the seasoned professional. I am Hannah Houston, and today I am joined by Dr. Gabriel Bossé. Gabriel Bossé is currently a postdoctoral fellow with Randall Peterson at the University of Utah. Prior to joining the Peterson Lab, Gabriel did his graduate studies at the University of Laval in Quebec City with Martin Simard, where he studied the regulations of the microRNA pathways with the nematode C. elegans. After completing his PhD, he was interested in developing novel behavior-based assays to study neuronal function and disease modeling. He chose to join the Peterson Lab with the hope of combining such assays with unbiased screening approaches, such as small molecule screening. Given the ongoing crisis with opioid abuse, he set out to develop the first self-administration model using the Zebrafish model. This unique assay allows him to study the impact of different small molecules on opioid seeking and to investigate important biological pathways regulating this behavior. Welcome, Gabriel. What an interesting area of research that you've chosen.
Thank you. Thanks for inviting me.
So can you tell us a little bit more about your research? It's very fascinating.
Yeah, so really the goal of my research is to use zebrafish as a model to study both how the brain works, but also to model neurological disorders. So for example, addiction is one of those disorders, but also interested, in anything particularly relevant to impulse control, aggression and things like that, which these symptoms are part of different disorders, including addiction. And really the goal is to use zebrafish both to understand the biology controlling these events, what are the pathways involved in aggression and impulse control, but also why do we have symptoms of hyper-aggressive person or somebody who have a lack of impulse control, or in the case of addiction, what are the pathways that regulate self-administration. By understanding the biology, we can then understand and perhaps create new treatments. And by using small molecule screening, that's one of these approaches that you can have molecules that modulate that behavior. So for example, you have a molecule that can affect how much an animal consumes an opioid, and then that can give you a lead for potential treatment.
Wow, what a fascinating area of research and so important, especially right now. Can you tell us a bit more about why zebrafish are particularly well suited for researching opioid addiction?
So I think, zebrafish is an emergent model for neurological processes. And I think it brings a set of tools that are not necessarily present in existing models. Like rodent models have been the primary model to study drug addiction. And they've been making great progress in understanding how the brain works and how the circuits are changes and implicated in addiction. But I think zebrafish brings a set of tools, like I mentioned, that are different. It's really a strong genetic models. For example, you can easily do mutants using techniques such as CRISPR-Cas9, and then you can then study how that affects the process of addiction, for example. And really, one of the key points too is the ability to do large-scale assays and large-scale approaches. So my assay, for example, you can train fish fairly quickly to acquire the self-administration conditioning, and you can train a lot of animals in a short span of time. And then again, when you talk about small molecule screening, with zebrafish, you can put molecules directly in the water. So no need for injection. So it's fairly fast. And then you can test a lot of molecules fairly rapidly. I think that's a key strength of zebrafish, the ability to do those large scale assays. That it's much harder and importantly, very expensive to do with rodent. So you can't do these assays in fish. So for me, in my view, I see fish as a complement to existing models. It brings a different set of tools.
Yeah, we are really big fans of the multimodal approach as well, and believe strongly in the fact that every model can provide different benefits to your research.
Yeah, I think so. Definitely.
So in your research, do you use larvae or adults? And is there one you prefer over the other for your research?
Yeah, that's a very good point. And I think it comes down to what you want to do exactly. What do you want to study? What are you trying to measure? Because both adult and larvae have sets of strength that are different. So if we think about larvae, it's simple, small animals, so doing large scale approaches are much easier. You can think about, for example, if you just want to look at locomotion, let's say, you can use like a 96 well plate and test like, different mutants, different molecules, and very rapidly you can go through thousands of molecules, dozens of mutants. You can study a lot of different parameters or changing different conditions. And again, if combining that with CRISPR-Cas9, for example, looking at the development of the animal, looking at how these different mutants affect how the animal behaves, larvae are very well suited for that. And the other aspect is that larvae are translucent, meaning that you can image anything, almost anything in the larvae, whether it's like the heart, how it's beating, how different cells might be expressed in the heart, but also you can go down to like brain activity. There's some very cool experiment that has been done over the years to look at how the brain is reacting to changes in light and changes in the environment in real time. So in an animal that is living, and you can see the entire brain, how it's you know lighting up, how the neurons are communicating in a live animal. So larvae are great models for that. But adults bring like the aspect of complexity. Adults can do much more complex tasks or complex behavior. For example, you think about social interaction, you think like memory formation and things like that, or in my case, the opioid self-administration, that larvae don't have the sets of skills to acquire these more complex paradigms. So, you can still do high throughput assays with the adult animal, um, not quite as much or as large as larvae, but really depends on which type of experiment you're trying to do and what are your takeaway you're hoping for. So you know you, larvae at 7 days old, for example, are not social animals. This is not something that they have acquired yet. So if you're interested in social interaction, then you go with a bit older animal. It comes down to the biological question that you're asking, I think.
No, that's such a good point about the visualization aspect of the
Mhm.
larvae that it makes everything so much easier when you
Yeah.
can see it with your own eyes.
Mhm.
Um, what assays do you use in your studies?
Mhm. Yeah, so the main assay that I'm using is an opioid self-administration. So basically, we have a rectangular arena with two platforms, and then we train fish to swim towards one particular platform that we call the active platform. And if they go to this platform, they receive a dose of opioid. So we use the opioid hydrocodone. Um, so it's one of the most commonly abused opioids. It's used in clinic extensively. Um, so fish essentially trained to go back to this platform to get a dose of drug. And then we can measure the number of time that these animals swim to at this platform. And then it gives us a readout on opioid intake or consumption, if you want.
Hmm.
This is the main assay. We do that with adult. I was talking about the complexity. They have to learn that they have to swim to that location, and then they have to form associative memory. So associated this particular platform with receiving a reward. So that's something that larvae could not do to that extent. So this
Wow.
is the main assay I'm using, but on the side I've also developed or optimized assays to look at memory, so like novel objects, for example.
Mhm.
So we can look at memory, stress and anxiety. We can look at social interaction. All of these with adults, for example. With larvae, as I mentioned earlier, you can do large scale approaches with like simple behavior, but you can still look at like sleep. You can look at stress and anxiety. You can
Mm.
look at like baseline locomotion, um, and also you can do some like non-associative learning that was called habituation. So if you repeat the stimuli, like they're gonna stop responding to it as they get used to it, if you want. But
Ah,
you have so that's one of the assays I'm using. So like it's really a battery of assays, like a platform that you can use after that to understand how a compound, whether it's a natural molecule, whether it's a molecule used in a clinic, how is it affecting behavior. So you're going to get the signature of the behavior and
mhm.
try to understand how does this change. So, and after that, you can also use such platform for mutants as well. So
Right.
understanding what sets of behaviors are affected by the treatment, the mutant. Um, that these are very useful tools.
Wow. Yeah, no, that sounds like a really interesting approach. So can you tell us a little bit more about why you choose a small molecule screening approach in your research?
Mhm. Yeah, so that's one of the key strengths of the Peterson lab. So Randy has been a pioneer of small molecule screening in zebrafish. And for me, what interested me initially was the unbiased aspect.
Hmm.
So let's say you take a library of like 1,000 molecules, it could be, let's say, natural compounds. These are compounds that we don't know necessarily what they're doing. And really, they're gonna maybe lead to the discovery of, first new biology. They can target a pathway that you have nobody suspected that it could be involved in this particular biological process. But also, it could tell you that this molecule could be useful to treat this condition or to improve a particular mutant, something like that. So really, for me, the unbiased aspect was the key. In my PhD, I used a mutant that was isolated from a genetic screen. So it's a different unbiased approach.
Right.
In that case, it was generating random mutation and I identify mutants with this particular set of phenotype. So and so really I got interested in small molecule screening as like an alternative to genetic. It brings different sets of, again, strength and a different tool than genetic screening. But really the aspect that you can treat, sets of molecules. So it's not that you can have it hypothesis driven, but the fully unbiased as well. Then I think it can lead to exciting new discoveries, exciting new biology that really were unexpected.
Right. Yeah, removing biases from our research is it can be difficult, but it is crucial.
Mhm.
What are the next steps for your research?
Yeah. So one of the next step, and it's something that we did with the latest discovery. So we identified a compound in zebrafish called finasteride that affects opioid consumption. So you treat fish with this compound and they reduce opioid intake. And one thing that we wanted to do is to validate these findings in rodent. So that was one of the steps moving forward. Is it conserved in rodent? So that's something that is interesting. Validate. Again, you mentioned multimodel approaches, so that's one way to do it. And really, so that discovery with finasteride, I then opened the door as well to study the role of neurosteroids in opioid addiction. So this is a molecule that affects neurosteroids production. So, investigating downstream pathways affected by finasteride. What is the biology of it? How is finasteride reducing opioid intake? So that's one of the, moving forward, that's something I'm interested in studying. But also, we're looking at potentially small clinical trial even. Can we use the discoveries that we made in fish and in rats? And could we improve quality of life of people, of like patient affected by opioid addiction? So that's one of the key thing. What can we do with those discoveries? But also, how could we improve the assays? How can we improve zebrafish as a model, both for addiction and also as platform for studying neurological disorders?
Wow, yeah. I mean, opioid addiction is such a
Hmm.
huge problem right now,
Hmm.
and your research really could have a huge impact there. So it'll be really exciting to see where it goes in the future.
Yeah, we're very hopeful that it could, even if you improve the quality of life of a subset of patients, it'd be amazing. That's
Right.
kind of the dream for a scientist.
Oh, absolutely. Well, your work focuses on zebrafish. You validate in rats.
Mhm.
Can you speak more on the benefits of a multimodal approach and
Yeah.
why you choose to include this in your research?
Yeah, and I think we already discussed briefly about it, right? I think different models have different strengths, and they can complement each other nicely. And also the zebrafish to study addiction is still like a new field of research. And it's still something that we're trying to establish as a robust and validated approach. And as I mentioned, rodents, especially rat, have been the gold standard in addiction research. I mean, yeah, obviously you can use non-human primates, but there's a set of complexity there as well. But rat has been really used over the years. an extremely powerful model to study opioid addiction or addiction in general. And really, our goal was to use like validated approaches, like rat self-administration. There's established protocols, there's established ways of doing it um, that has been used for years and years. And really, by validating that the compound that we identified in fish also works in rat, gives strength to the story. We know that finasteride can reduce opioid self-administration, for example, in different models. Animals treat condition with different protocols, and I think it really shows that there's potential for translational research. And any research that has a translational aspect often goes to rodent in the steps of understanding. There is more and more molecules that are going from fish directly to clinical trials. But in the field of neuroscience, which is still an emerging field for zebrafish, I think it's still, often people go to rats with validated approaches. So for us, that was important to confirm that what we found in fish was conserved in mammals.
Yeah, no, that's really unique and wonderful that you're choosing to take that approach.
Yeah.
Now, for our final question, zebrafish have been slow to gain popularity in drug discovery research compared to other models such as rodents. Do you think the field is changing towards more adoption of the zebrafish model?
Yeah, I think so. And I think fish is being, like I said, validated more and more. So meaning that the field is accepting fish as a solid model to study in neuroscience, let's say. And I think it comes down to appreciating that fish can have like a complex range of behaviors. There's that old saying that, goldfish has a 2-second memory, something like that, right? And it's not true. Zebrafish in particular are showing, that they can establish memories. They can, they react to the environment. They show sign of anxiety. They are a social animal, so there's a complexity of behavior there that is being more and more appreciated. Like I mentioned too, it brings a set of tools that is different from rodent, and there's also, a push to more cost-effective methods to study different like disorders, to do research in general, right? And also to try to reduce our use of animals, especially in mammals model. And I think zebrafish can bring a lot of like extremely powerful methods that can, complement and in some cases replace rodent model. But it's also tricky to say replace, because I think it's complementing what's existing. But I think, it's a cost-effective method to study some of these disorders, bringing a different set of tools. And I think that people are starting to appreciate and understand the validity of using zebrafish and also like how powerful that can be.
Right. Yeah, and I mean, we at In Vivo love zebrafish, so we are very excited about that and the trends of towards more zebrafish
Mhm.
adoption and popularity.
Yeah. Yeah, I think it's a great model.
Well, that's our time. So thank you so much for joining us today, Gabriel. It was wonderful to hear more about your research and about zebrafish in general.
Yeah, it's my pleasure. It was great to be with you. Again, thanks for the invite.
Well, thank you so much. And to everyone, tune in next time for 17 Minutes of Science. Bye.
See ya. Bye.